Hello everyone, my name is Mark Kuo. I am currently a general biology major as part of the College of Math and Natural Sciences at the University of Maryland, and I am also one of the life science scholars that are currently here to present my academic showcase poster. To start, I guess I should give a bit of an introduction into the topic of oncogenic pathway inhibition in cancer research. Cancer research has only really come into fruition within the last 100 years, and as of now, common treatments include surgery, chemotherapy, and hormone therapy. However, drug inhibition of oncogenic pathways has become a new method of cancer therapy, and it is what my fieldwork site, the Solid Lab, focuses on. The Solid Lab is part of the Zuckerman Research Center at the Memorial Sloan Kettering Cancer Center in New York City, and it mainly focuses on tumor-causing genetic alterations and pathway inhibition. There, my main objectives were to, at first, obtain basic wet lab skills and knowledge of the cancer research field, but also to contribute to existing efforts to find viable oncogenic pathway inhibitors, or basically ways to inhibit cancer growth or the spread of cancer within the body, but also to, in the end, provide data and cell lines for future lab use and for future research to build on, since I was only an intern and I would only be there for a short period of time. My main methods of data collection involved Western blots to measure levels of protein expression in cell clones following drug treatment, since the Solid Lab greatly believes in the power of Western blots and the amount of information they can provide you with. I also treated cells with kinase inhibitors for identification of pathway targets, basically running through different drugs to find which ones best inhibited or killed cancer cells without killing off normal non-cancerous cells. I also performed cell viability tests using MTT assays to tell how many cells were killed after drug treatment, and also, in general, I just cultivated and maintained cell lines for use in oncogenic inhibitor trials. Mainly, the results that came from these experiments are more or less have to remain private due to the overall the overall area I was working within. I can't really say much about the type of drugs or the cell lines that I was using, but I can say that two of the drugs tested provided somewhat promising results. While the doses do probably need to be adjusted from this point on, I believe that in the future they could become useful in inhibiting parts of the oncogenic pathway. And it also opens up the possibility of combining other inhibitors with those drugs to have better effects. My overall experience was greatly positive, and my knowledge of basic lab techniques has greatly increased. I also have a better understanding of Western blots and how genes affect cancer treatment, and I'm also more up to date on current efforts to treat cancer, since to start, I didn't really have much knowledge of the cancer field at all, and if anything, I'm just glad that my knowledge has increased and that I was able to meet such amazing people. And with that, I just wanted to thank my mentor, Michelle Tang, Dr. Paul Gao, Dr. David Solit, and the rest of the members of the Solid Lab for this meaningful experience. Thank you.